Hello and welcome to Quarantine Lab. These are my lab assistants, Chloe, Caitlin, and Trent. Today we are covering Chapter 10 about muscles. We're going to begin Chapter 10 by having Caitlin lift different weights that will be handed to her by Chloe. All right, so go ahead. So what you can see happening here is when Chloe put the heavy weight in Caitlin's hand, that her proprioceptors and her muscles and her elbow were able to feel that it was a heavy weight. So her brain knew it was something heavy, and her brain knew to stimulate several motor units in order to lift that weight. With the lighter one, her proprioceptors felt that it was something light, and her brain knew that she didn't need very many motor units to lift the lighter weight. Now we're ready to do the part where someone stands in a doorway, and Chloe is going to demonstrate this for us. So what we're going to do, go ahead, Chloe, into the doorway here. We're pretending that there's been an earthquake and the building is falling down, and we're relying on Chloe to hold up these walls. So go ahead and put your hands out. Now you've got to use all your strength. So what's happening is Chloe is trying to contract her muscles. Her nerves are sending signals to her muscles, which is causing the calcium gates to open. Calcium is being released. The calcium is binding troponin, which is moving tropomyosin, and she's forming cross bridges. Now remember, the next step after cross bridge formation is power stroke. But because she is in a doorway, these walls are physically preventing her muscles from doing the power stroke step. So she has lots of calcium being released, she has lots of cross bridges forming, but no power strokes occurring.
5, 4, 3, 2, 1.
So Chloe experienced her arms floating up, even without, you weren't even really trying to have them float up, were you? So that's because you get all that calcium, all those cross bridges. Once the wall is no longer in the way, now the power strokes can happen, and your muscles contract and your arms raise up. Now we are ready to cover the length-tension relationship, and we're going to fill out the chart on this page. So Trent is going to demonstrate for us the strength in the 3 different positions. So we're going to start off. Trent will have his wrist in the extended position. Go ahead and squeeze. And there you can see he was able to raise the needle to 65. All right, thank you. Now Trent will put his wrist in the hyperextended position. Now in this position, these muscles right here are stretched. So now he's demonstrating the picture here with the muscles stretched out. In the first one where his wrist was extended, that was the normal one. Okay, go ahead, Trent. Remember, don't straighten that wrist. You gotta keep it hyperextended. Okay, let's see your reading. All right, hyperextended, his reading is 30. Okay, now he's going to demonstrate with his wrist in the flexed position, which is all scrunched up. So now with his wrist like this, these muscles are scrunched up. So they're this top picture. All right, go ahead.
Oh, wow.
It feels really weird doing it in that position. And your reading, could you hold that upright? Thank you, is 10. All right, thank you, Trent. We're going to start off with the basic structure of a muscle. In a muscle, at the most microscopic level, you have these long strands of protein. The 2 main ones are actin and myosin. So if we pretend that these red and yellow pipe cleaners are actin and myosin, this is a representation of the inside of a muscle fiber. The muscle fiber has a cell membrane called a sarcolemma. So this pink and black cloth represents that. So muscle has long strands of protein, the actin and myosin, and that is wrapped up in the sarcolemma, the cell membrane. Then you take a group of those and you wrap those up together. So individual fibers are wrapped up in endomysium, so you would have a layer of endomysium outside of this. Then you take several fibers. So here you can see individual fibers. The pink represents the individual fibers, each wrapped up in its own sarcolemma and endomysium, and you wrap those up together in perimysium. So the green represents perimysium. Now you have a bundle of muscle fibers in perimysium. This is a fascicle. Then, if you take several fascicles, so each bundle of green here represents a fascicle, and you wrap those up together in another layer of connective tissue called epimysium, then you have an entire muscle. So this bundle represents an entire muscle. It has epimysium, which is the tan. Inside of it, you have these units called fascicles, represented by the green. The outer layer of the fascicle is perimysium, which is this green sheet. If you unroll one of those, you find within it individual muscle fibers, the pink, each wrapped up in endomysium. Now let's come over here and look at these models. This is a model of a muscle fiber. So this is showing us a cell of a muscle. Remember, fiber is another name for a cell. Within it, you have units called sarcomeres. So from one red line here to another red line is a sarcomere. And you can see how these all line up end to end. In your book, it describes it like a train, how they're all lined up end to end, are the sarcomeres. The blue part here is the sarcoplasmic reticulum. That's where the calcium is stored. And this purple is the T-tubule, and here's your sarcolemma, the cell membrane. So the cell membrane will receive a signal from a neuron. The neuron's going to secrete acetylcholine onto here. That'll cause an action potential on the sarcolemma. The action potential, which is an electric signal, will travel down the T-tubule. That'll stimulate the sarcoplasmic reticulum to release calcium. The calcium will come out here, and it will bind to troponin, move tropomyosin. The actin and myosin can form cross bridges, and these red lines are the Z-disc. They will get pulled closer together. And if each one of these things, each unit here, each sarcomere, has its Z-disc pull in toward the M line, the sarcomere gets shorter. And when all of these get shorter, the whole fiber gets shorter. And that's a muscle contraction. So this model is showing us an individual fiber. It would be like what we have here in the pink with the actin and myosin inside. We're looking at that up close. These green lines are the myosin. The purple lines here are the actin. This model is a sarcomere. So we're taking here what we have from one Z-disc to the next, this individual sarcomere, where you have hundreds of them lined up end to end in a muscle, and we're looking at it up close. So the yellow lines here represent the T-tubules. That's what carries the action potential down into the muscle fiber. The teal is the sarcoplasmic reticulum. That's where your calcium is. We take this off, we can look in here. Here's the M line, the center of the sarcomere. Here's a Z-disc, here's another Z-disc. That's the ends that are going to get pulled in closer together. The blue here is the myosin, and you can see the myosin heads. The red is the actin. The yellow stripe is the tropomyosin, and the green is the troponin. So when you get that electric signal from the neuron, so the neuron secretes acetylcholine onto the muscle, that causes an action potential in the sarcolemma. The action potential comes down the T-tubule. When you have an action potential in the T-tubule, the sarcoplasmic reticulum releases its calcium. That calcium diffuses out and it binds to the troponins, the green things here. Once the calcium binds to the troponin, it moves the tropomyosin, the yellow stripe, out of the way. Notice here the myosin heads. See how they want to come up and connect to the actin. As long as tropomyosin is in the way, they can't connect to the actin. Once the calcium binds to troponin and moves tropomyosin out of the way, now the myosin heads are free to bind to actin. So that's what happens, and that's called cross-bridge formation. The myosin heads bind to the actin. Same thing will happen on this side. The myosin heads will bind to the actin. Then you have power stroke. These myosin heads bend. So they can stand up and they can bend forward. So once you get the cross bridges, the myosin heads bend and that pulls the actin in. Think about like pulling in an anchor on a boat. You have to keep grabbing the rope and pulling over and over. So then the myosin heads will let go, they'll pop back up, they'll bind, and then they'll bend again. Each time they do that, they pull the actin in a little bit. So the actin gets pulled in little by little toward the M line. Same thing happens over here. It gets pulled in little by little to the M line. Then you have your sarcomere shorter. So when you started out, the sarcomere was like this. You had this gap here, this open space. Notice your Z disc here, your Z disc here. Look at the length of the sarcomere. The calcium binds troponin, moves tropomyosin. You have cross bridges form. Then the myosin heads power stroke. This one gets pulled this direction. This one gets pulled this direction. The Z disc are now closer to the M line. The sarcomere is shorter, and that's how a muscle contracts.